Hello, my name is Stephan Sieber. I'm a chemist at Technical University in Munich. On the 9th of November 1989, I was going to high school, and in the evening I was watching television with my parents and a friend, and we were not believing what we were seeing. Incredible joy was with us. When I was a child, I actually didn't want to become a chemist because my grandfather and my father were both chemists. I wanted to become a pilot. However, that didn't come true because I grew very tall. I'm 6'8", 2 meter 03, which is too tall for flying planes. And therefore, actually influenced also by great high school teachers, I then decided to go the way into chemistry, and I don't regret it. Our research literally breaks the bacterial cell wall. So we are looking for new antibiotics that overcome the current resistance crisis. This crisis is due to multi-resistant bacteria that cannot be addressed by any antibiotic on the market. The situation got worse because antibiotics have been used in a wide range of applications and not always in their dedicated role. So this has led over the past decades to the accumulation of many resistant strains where we are currently looking for new solutions. A major limitation of the classic antibiotics is that they go after a very limited number of essential cellular targets, which is cell wall biosynthesis, DNA or protein biosynthesis. However, considering that there are hundreds of essential proteins in bacterial cells, the goal would be to look after those other essential protein targets and find selective and tailored drugs and compounds that switch off these or address these additional proteins in the cell. And this is the major point of our research, looking for unprecedented and novel pathways. In the search for novel antibiotics, we identified a small molecule by a screen, which has two unconventional modes of action. The first one is the stimulation, meaning the overactivation of a bacterial signal peptidase, which leads to the uncontrolled release of proteins outside of these bacteria. And we can show that this also breaks the bacterial cell wall. The second mechanism is the interference with energy metabolism by menaquinone biosynthesis. So another innovation or innovative aspect of this approach is that addressing two targets at the same time makes it much more difficult for bacteria to develop resistance. And in fact, we were not able to generate any resistant strains in the laboratory. Moreover, the compound is effective against dormant persisters. Those are the ones that are very difficult to treat and to combat, as well as biofilms, which are a problem on canthethers and stents. So taken together, these innovative aspects of the mode of action, together with the properties, we think that PK150, that's the name of the compound, is a worthy candidate for further development. I think an immediate benefit for society from our research would be the development of a clinical candidate that has the potential to become a new antibiotic drug, which is currently really desperately needed due to the resistance crisis. However, a more indirect implication would be to stimulate research by innovative ideas in this area and make more efforts towards the development of novel antibiotics, which has been neglected for a long time. Development of our compound is at a very early stage, and yet it is not clear if it will be effective in a human being. This question, together with issues of toxicity and other side effects, sometimes keep me lying awake at night. When I talk to a friend about his research, she always tells me that she knows people directly affected by bacterial infections. In some cases, they were even very difficult to treat. So her feedback is always that research is, of course, something that you can easily understand, and also the impact for the direct friendship and society becomes clear and doesn't need further explanation.